Good afternoon and welcome to this exciting presentation with Curasight. With me today I have the CEO of Curasight, Ulrik Krasilnikov, together with the CSO, Andreas Kjær. And just some practical issues before Ulrik and Andreas take us through the 2022 highlights and update on the pipeline. Today's event is in English, and please post your questions either in Danish or in English on the right side of your screen. So with that said, welcome Ulrik and welcome Andreas.
Thank you, Claus. Thank you for the introduction. We have prepared a presentation for you covering the most important parts from the last year and also what we expect to reach for the coming year. Just a short disclaimer. The agenda will cover today the financial results from the 2022, and then we'll cover some highlights during 2022, and then we will highlight some what is the current focus for us, and then we will dig into also what are the milestones we expect to reach this year. The financial results covers both Q4 and the whole year of 2022. I will only cover the whole year in this presentation as the time is limited. But as you can see, we still have a net sales amount to 0, meaning that Curasight is still a development company, so we don't have any sales yet. The operating loss amounted to around minus 19 million Danish crowns compared to 11.4 last year, meaning that we have increased the development activities. If you look at the loss after tax, it's around 18.3 million Danish crown compared to 11.1. So I would say in general the results are reflective of the increased development activities, also due to the expanded strategy. The total assets amounted to around 60 million Danish crowns compared to 79 million last year, meaning that we have used some of the cash. Still, we have a very good cash position, which sums up to around 50 million compared to 73 last year. If you look at the ratio, equity ratio, it's around 95 compared to 97.4 last year, which is still very satisfied. So that is not that is the main case I would highlight right here. Then some highlights during the last year. In the beginning of January last year, we reported some results from the investigator-initiated phase 2 study performed by researchers at Rigshospitalet in U-Trace, where we reported the results from the study in neuroendocrine tumors, which demonstrate a very strong prognostic value and also the potential future of use in radionuclide therapy, meaning U-Treat. Furthermore, in the same month, in February, we reported that we have acquired the early-stage company TRT, which also strengthens our therapeutic platform and also the development in that path going forward. And furthermore, in February last year, we reported that we want to expand the clinical program in 2 additional indications in neuroendocrine tumors and head and neck cancer, which is based on the positive phase 2 results from those studies in U-Trace. And in April, we reported that we have granted a patent, US patent, also within U-Trace, but based on the copper agent 64 copper, meaning that U-Trace will cover both gallium-68 and also copper-64, but Andreas will come back to that. And then it was reported that the investigator-initiated phase 2 study in brain cancer with U-Trace was completed. The inclusion was of a patient was included by the end of Q2. Based on that, also we initiated a preclinical studies in both in neuroendocrine tumors and also in head and neck cancer. And that is based on the previous announced positive results from the phase 2 studies in U-Trace. But we expect also that the preclinical studies will be reported here in the first half of 2023, and that is also based that we want to pursue the theranostic principle in those two indications. Then just not specific, but SEB also made an update of the commission research based on the expanded strategy, also that they changed the range valuation range of the share of Curasight. So it covers around 45 to 63 Danish crowns per share, with a midpoint of around 54 Danish crowns per share. But we expect also that that will be changed during the year, this coming year, when we look into the milestones we expect to achieve. But more about that later on. Then I will hand over to Andreas, who will take you through the current progress.
Yeah, thank you very much, Ulrik. So I will update you on where we stand with the pipeline, and of course, for those not necessarily that have not necessarily attended previous presentations, also give an overview. So our technology, and I come back to that, is unique in the way that it's not limited to one cancer type. It's actually present on 80% of all solid cancers. So we have the luxury of being able to select which cancer types we are pursuing, and of course we are picking these and choosing these based on the unmet medical need and the competitive landscape. And of course, finally, our bandwidth, because we cannot pursue in all cancers at the same time. And what you see up here is that our main focus has been from the beginning and still is on brain cancer and prostate cancer. And in prostate cancer, for now, we are pursuing it as a diagnostic indication, so that's with uTRACE only. Whereas in brain cancer, we are pursuing it as both imaging and therapy. I come back to that, how that works in synergy. And then based on the studies, as Ulrik mentioned, in neuroendocrine tumors and in head and neck cancer that were undertaken, investigator-initiated studies at Rigshospitalet with very positive results, we added those two indications to our pipeline. So we are pursuing in both these indications also both therapy and imaging. So to go a little bit then into what we are pursuing with, then our proprietary theranostic platform is made up of a diagnostic agent called uTRACE and a therapeutic called uTREAT. And together this is called a theranostic pair. And the nice thing is that since these target the same molecule, namely uPAR, you can use the diagnostic to see whether therapy is worth pursuing. So in other ways, if the imaging tracer is not getting into the tumor and sitting there, then the therapy will also not get in there. But I'll come back to that. The target we are pursuing is called uPAR, which is an abbreviation of urokinase type plasminogen activator receptor. And this is an extracellular receptor, which means it sits on the surface of the cells, on cancer cells, on the invasive front. And the more aggressive the cancer cells are, the higher it is expressed. And this is important because when we target with radioactivity with uTREAT, the cells that are uPAR positive, we get the most radiation to the most aggressive cells. So in this way, it's an intelligent way to distribute the radiation within the tumor. Then it's low expressed in healthy tissue. And again, as I mentioned, not mentioned to one type of cancer. So we can actually pursue with once we have developed the agents, they can be used in many types of cancer. Again, just reminding what is target radionuclide therapy, you can say it's basically injectable radiotherapy. So instead of what you see on the left, where you irradiate with a radiation beam from the outside, this is traditional radiotherapy, you have to shoot through healthy tissue in the way to the tumor. with radionuclide therapy, you inject the ligand that is radioactive and that binds to cancer cells. So it distributes, it seeks out the cancer cell, it binds, and from there it irradiates. And the type of radiation we use has a range around 1 millimeter only. So in this way, it's not irradiating healthy tissue, but only the cancer it's sitting on, or mainly the cancer it's sitting on. And in this way, targeted radionuclide therapy is more gentle than external radiation therapy. And many believe, including myself, that maybe in 10 years, this will be the main way to do radiotherapy, much more than external radiation therapy, because it's more gentle. Then, as I mentioned, we have actually several indications we can use this principle in, but our number one indication is glioblastoma. It's an aggressive brain cancer, 30,000 new each year of the aggressive type in the US and EU, and unfortunately 10% of the patients are children. Why we are pursuing it in these tumors is that nothing happened over 15 years regarding survival in these patients, so they still only live around 1 year from diagnosis, and only 1 in 20 patients will survive 5 years. So there's room for improvement, and we believe that with our U-treat principle, we will be able to be a game changer in this very aggressive disease. And how does it work? As I mentioned, the companion diagnostic concept, or the theranostics, it's based on what you see is what you treat. And what you see here is, on the left, you see an MRI scan, and the white tumor in the middle is the glioblastoma. On the right side, you see a patient that was injected with U-trace, and the red hotspot is actually where all the radioactivity sits. So in this imaging, or this image, you can see, yes, when we inject U-trace, it gets access and sits in high concentration in the tumor. By showing this, we also know that the counterpart U-treat, because it is the same peptide binding to the same receptor, will also sit in the same place. And therefore, this is a patient where we will probably have a good efficacy. Reversely, you could say, if we did see nothing light up on this in this patient, it will be a patient we will not pursue for treatment, because we can see the drug is not getting there. But the drug is in the vast majority getting there. That's at least what we know for now. This is just a diagram showing a little bit what I showed on the first part on the principle of targeted radionuclide therapy, but just for the brain cancer. On the right, or in the middle, you have how targeted radionuclide therapy works, for instance, with U-treat, where we inject it and it sits then in the tumor because the tumor expresses UPAR. On the left, you can see that external radiation therapy is done by irradiating from many different angles. On this diagram, they are shown 3 different angles. They always include the tumor, so thereby we get an accumulation in the tumor. But all the green and yellow and orange area is healthy brain tissue that is also irradiated. And for this reason, you can easily understand that the targeted radionuclide therapy is a much gentler principle. On top of that, it can also treat disease that has spread. If there are metastases or small tumor deposits, what you inject will also seek out these cells. You cannot do that with external radiation therapy because these deposits cannot be seen on MRI. So in essence, you do not know they are there. So therefore, we really pursue on the theranostic path brain cancer. If we then go to prostate cancer, and for now we are only pursuing imaging here, then the large problem in prostate cancer is that the majority of the cancers are not very aggressive. So when I went to medical school, it was always said more people die with than of prostate cancer. The problem is that it's true, but not true for everybody. So you are unhappy if you were not the person where it was a benign disease. And the major problem is well recognized to be in these tumors, to find out which tumors will stay benign and therefore should not undergo aggressive treatment. Today, this is followed by biopsying patients regularly. This is not very gentle. It's painful. There's bleeding and there's infection in many patients. And on top of that, the technology is quite unreliable and very unreproducible. I can mention the good example, a study that showed smaller tumors found by biopsies, and then they were re-biopsied 2 months later, and then 30% had a readout that they no longer had cancer. Of course, they still had cancer, but the biopsies just missed to biopsy from the cancer. So this demonstrates how unreliable it is. We want to implement instead a non-invasive biopsy, which is U-trace, so you can do a scan. You do not overlook anything because you see the whole tumor. You are not relying on where you biopsy. And in this way, much more people can be followed in what is called active surveillance. This is important because today, with current ways to do it, people are overtreated because the physicians think better safe than sorry. They know the biopsies are not very comfortable, but also not so reliable. So they say, well, it might not look so aggressive, but better remove the prostate gland because then nothing happens. And is this trivial? No. 70% of patients getting their prostate gland removed will experience some degree of impotence and or urinary incontinence. And if we then move forward to how this looks in patients, and we have already performed this in some patients, then you can see in this image the CT scan to the left. Between the red arrows is not the tumor, but the prostate gland. And in the middle, you can see the tumor light up, and on the right, with U-trace, and on the right, you can see the two things fused. And this is then a patient that lights heavily up on the scan, and therefore a patient that needs treatment. If it had lightened up only very little here, then we would have a cutoff, and that's what we want to establish. So we could say, you are safe, you can just come for another scan in 1 year. Yeah, and so Ulrik, this was a little bit about the technology.
Thank you, Andreas.
Could I just pose a question very quickly, Andreas, just to, if it's okay with you, Ulrik, just to
yes, please, feel free.
What you do with prostata,
yes,
Andreas. So in the case, it's not a severe case. It's a severe prostata where you have to take the use of surgery. Would you enroll the patient like you do with mammography in breast cancer? So after 1 year, you scan again, and maybe 1 year more you scan, and maybe you don't even see aggressive development during the man's lifetime.
No, exactly. You are right, although not 100%.
Okay.
So it is exactly the same concept. We are not at a screening level. So the only difference to the breast cancer strategy is what you do here is that these are patients where we already know there is a localized cancer in the prostate. And then we have to find out and follow whether it develops into some aggressive cancer. If you take what I'm sure you're thinking of mammography screening.
Yes.
In that case, you are actually screening for whether there is a cancer or not. So this is the difference. The difference here is that the inclusion criteria for getting a U-trace scan is that we know there's a tumor there, and it's most likely benign and will not develop, But instead of starting with biopsies and maybe over-treating and taking the prostate out without a need, we just make a scan and make a readout of how aggressive it is. And if it's not aggressive, which is the most likely thing, then we will follow it with renewed scans every year or every other year. So the concept is the same, but it's not the primary diagnosis of the disease. It's the follow-up of the disease, whether it becomes aggressive. And this is special for prostate because the majority stays benign. If you detect a breast cancer, you need to remove it. There's no such thing as leaving a breast cancer behind. And this is because breast cancer, on average, is much more aggressive than prostate cancer.
Thanks a lot. Yeah, the reason I also ask is breast cancer and prostate cancer is the two major cancer types in the Western world. So it's very interesting that you're able to, with less, actually find more. So thanks a lot. So please carry on, Ulrik.
Thank you. Yes, and this is just to emphasize that in connection with our expanded strategy last year, where we announced the two additional indications, we also want to pursue the strategic partnerships, and that is actually something that has going on for the last 6 months, and which we hope that will materialize this year within 1 or 2 or more. It's not that fixed to say, but we hope that we will be able to enter at least one agreement or more this year. But it's the reason we are pursuing those partnerships is, first of all, that it's a validation of the technology, and it by that also de-risk the case. And furthermore, we will get a non-diluting funding, which is also very important in the times right now. And furthermore, we get the power of speed to market, as we are mainly a development organization. So it's very clear, and also not only for us, but we can also see other companies are now pursuing this track by entering partnerships. So from investor point of view, it will also be a good thing when it's possible to announce that, hopefully.
Ulrik,
in the near future.
Sorry, just a quick question here, because you have a very remarkable list of partners on this slide. Have you entered into partnerships with all of them, or are you in negotiations, or how far are you?
This is only prospects, so to say. So, but I cannot be that too specific, but because we expect that to be very positive value inflection point for the company in general. But these are all companies dealing within this nuclear field that we are also dealing with, and they have the power and also the organizations to take products like uTrace and uTreat to the market. So that is, let's say, companies that
Good. Do you hear me, Ulrik?
Then we will look at the milestones.
Yes. Thanks a lot, Ulrik. I think you disappeared for like 30 seconds. So, but you finished answering my question on the partnerships. Of course, it's always a little sensitive, and I know if you enter into a partnership with a milestone, you have to press release that. So of course we look forward to that. Do you want to take the milestones first, or should I shoot a couple of questions?
You're welcome to shoot a couple of questions.
Yes, because there's questions coming in here, just so we manage them the right way. So there's a question here that's probably to you, Andreas. You finished the phase 2 study of uTrace in brain tumors the autumn of 2022.
Yes.
Now we are waiting for the final readout. And if I compare the slide from Q3 to the current slide, it looks like there's a small delay. First of all, of course, if you could comment on that. And secondly, how are you going to design your phase 3 study? And what comparison group are you going to use?
Yes. So this is a very good question. And first of all, it's correct that the reporting of the results from the phase 2 in brain cancer is a little bit delayed relative to what we have said. The important thing here is to understand that this is still an investigator-initiated studies by Rigshospitalet. So it's not Curasight that controls the timeline. We officially have been, and that has also been published, that all patients have been recruited, and that was actually not a thing that was changed. That was actually, I think, one of the days, last days, I think it was June 30th, actually, of first half year, where the last patient was included, and the readout simply is we simply have to wait until they get it published. So because it's not a company's study, we do not know the data before they have made all of them and have sent it in, and that is a process that typically takes some months. If this was one of our sponsored studies that will come in the future, we could tell you more precisely that we are at this step, but it's because it's still an investigator-initiated study. So this is the reason. So the short thing is, in contrast to a lot of other studies, we have actually been quite fortunate not to have seen further delay on the inclusion of patients, but now we are waiting for the reporting, and that could be in 1 month or in 3 months. That is that's the whole process also with getting it academically published. It can go back and forth one time or several times. So in my view, I don't see it really as a delay, because the data are the patients are that's normally the number one obstacle in any clinical trial, that actually to get the patients in, and the patients are in, and all the data have been secured, and now they are being analyzed and reported. So I would not look at this as really any critical delay. Then the question is about how our phase 3 study will be designed, and the phase 3 study will be designed on basis of the phase 2 data. So when we know the exact data from the phase 2 trial, then our statistician can calculate on these numbers how big a phase 3 study needs to be. So the precise size we do not know. One thing is sure, it's a larger study than the phase 2 study, and that is typically the case. Since it's a very severe disease, unfortunately, and a lot of people will die over a relatively short time of observation, the study will not be very large because the follow-up can be shorter. If we were looking on survival in a cancer where the life expectancy was much longer, it would be either a larger study or a longer study. So it will not be a very large study, but the precise design will depend on results from phase 2. And then just if I can elaborate a little bit more on that, then the protocol, the precise design is always submitted to the FDA, the US authorities, at what is called end of phase 2 or pre-IND meeting, where basically we present our plan, and they either buy into it or ask for changes. It's still our decision, but we get a feedback on whether it's likely that we can obtain approval with this data package. And if they say we would like to see this added, then it's very wise to follow that idea. So there are 2 steps: the readout on basis of which we can make the exact design of our study that will then be submitted to the FDA for a meeting where they will get their feedback, and then we can start the study. That is the order of activities in that study.
Of course, and thanks a lot, Andreas, for clarifying that. So are you able to carry out this study, depending, of course, how many people you have to enroll at Rigshospitalet, or do you need to involve Swedish, Norwegian, or German hospitals?
Yes, typically it's, yes and no. We might be, I think it would be difficult at one hospital only, but it would also take too long. Typically it will be a study involving maybe 3 centers. On top of that, it's also normally a wish in the later studies, in the phase 3 study, that one wants to have more centers in. And it goes with the authorities wanting things to be generally applicable. So there's always the risk that if you only do it in one place, they are extremely good at using your technology. But if you divide it over several centers, if it's a multi-center trial, it kind of secures that different people can use the technology and you get the same value of the readout. So even if we could make a small study that could be included only at one hospital, we would still wish to include several centers. And when I say several centers, it's several centers in several countries. It should not be centers from one country only, in particular not in a small country like Denmark.
Good. Thanks a lot. And if I may, Ulrik, there's one more question here. It's just about, it's a follow-up on you, Trace. Well, we know you have pretty good results already, but how do you see the difference between the different indications? So prostata and we have brain tumors, and you're even further down the pipeline looking at other indicators. Do you see more or less the same indications and not so cancer dependent, or do you see variations? Is that something you can tell us anything about, Andreas?
Yeah, so I think that, Ulrik, I think, yes, there is one big line between prostate cancer and then the remaining cancers. In prostate cancer, it is this biopsy following this huge amount of patients having it. So you could say it's a diagnostic surveillance tool. In all the other indications, the main indication is the therapy. The uTrace is basically supporting and demonstrating that the target is there. So those are, you could say, targeted radionuclide therapy indications. So there's a line between there.
Good. Thanks a lot. And Ulrik, please carry on with the milestones.
It's okay. I think we have most of it. But just what we would like to highlight is that we expect that, as Andreas already mentioned, that the phase 2 study in glioblastoma will be published very soon. And then we also have the results, hopefully, from the preclinical studies in glioblastoma, head and neck cancer, and also neuroendocrine tumor. We also expect that to be reported the first half of this year. And then we have the clinical trial application for prostate cancer. That's more the regulatory part will be taken care of the first half of this year. And also when we look into the second half, 2023, we have the new drug applications for prostate cancer and also for glioblastoma we will work with. And besides that, I would say this is only the clinical trials related to the clinical trials. Then we have the business part, but that's more unreliable to predict what will happen there. But we have a very good feeling that something good will happen, will occur in 2023. So that's what I will mention right now here.
So
yeah.
So Ulrik, with the current cash on your books, and you have a pretty good bank account, if I may say so. How are you able, with those plans you have right now for the two phase 2 studies? Is that enough to take you through 2023, or and of course it's depending on partnerships as well and data and everything. But with the expectations you have as of today,
I would say the whole program and all the activities we are currently pursuing is covered by the current cash position. But we are also pursuing the partnerships track, and that will provide us with some non-diluting funding. So depending on also what they expect us to accelerate or whatever they prioritize, then we will probably need new funding of the second part of 2023. But that's something we also work with in parallel. But that will be behind hopefully one or more partnerships agreements. So that what I can say right now. But the funding right now will cover the activities for the current year. So we are not forced on the short term to seek new funding.
Good. Thanks a lot, Ulrik. And to flavor a little on your partner strategy, now I asked before, there's a question coming in here. If we look into the strategy, is a potential partner looking into a combination, so both you trace and you treat, or are you able to sign licensing agreements with one partner on you trace and another partner on you treat when you're further down the line?
Should I start and then? That is I would say a very good question also, but also for clarification, because some of the partnership companies we are talking with right now is only pursuing the diagnostic part, the imaging part with uTrace. Other companies are only pursuing the uTreat, the therapeutic path, and some companies are pursuing the combination, the diagnostic combination. And that is the puzzle we have to lay right now to find out what will be the best strategic fit for Curasight, both indication wise, but also in power. It's not a secret that it's a big companies we are talking with right now, and they are. It's tough to deal with those guys because it's very long decision make process that they have right now. But I would say we have a very good position to get the products to the market. But again, it depends of what we find is the best path within each indication wise.
Good. Thanks a lot, Ulrik. Do you want to add something further, Andreas?
No, I can only, I think it underscores the technology's broad applicability. So there's both a diagnostic part, there's a therapeutic part, and there's the part that we can actually work in several cancer types. And with this matrix, it's, I would say maybe, it's definitely likely that we enter more partnerships, and it's this patchwork that we hopefully enter, and it's this patchwork that we are currently looking into, and so one thing is not blocking for the other one, and understanding what the interests of the different players really are.
Thanks a lot, both of you. And just to the audience, if you have any further questions, please pass them on. Doesn't seem like that there's further questions coming in the chat. So with that said, both Ulrik and Andreas, thanks a lot for a very thorough presentation. It's really nice to follow you, and we look forward to hopefully the next event with your data. So with that said, thanks a lot for all the good questions, and thanks a lot for participating, Ulrik and Andreas. And everybody enjoy your day. By that, I will close the event.
Thank you.
Thank you very much.